Thanks very much, Belinda, and she'll take questions after the presentations too. And now I'm going to have Steve Altschuler come up and introduce Susan Shen.
Hi, I'm Steve Altschuler. I co-mentor, if you could even use the word mentor, with Susan, with Lonnie Wu. I guess I will start by saying 5 years ago, on a rainy December in San Francisco, two remarkable things happened. The first was that a DARPA program officer came by and knocked on our door and said, I've read everything you guys wrote, and I think you guys have a way to crack open complex diseases. But you're going to have to write a proposal, and you're going to have to think not just about cells, but animals and humans. And I was thinking, cells, animals, humans, what are those? But then the next thing that happened was Susan came and knocked on our door and said, hi, I've read all of your papers, and I really think that what you guys do would be really interesting, but I work on animals and cells, animals and humans. And I was like, oh, perfect. So over the next month, together, even before she had joined our lab or was even allowed to work in our lab, we worked together over the holidays, Christmas, New Year's, and we put together, I'd like to say a story, but a proposal for DARPA as to how we would apply the systems biology approaches in our lab and scale them up to actually things that are relevant to humans, in their case, warfighters, and test them on animals. And all these graphs that we drew on PowerPoint, they actually ended up being true, but not just because we dreamed them up. It's because Susan came and she taught us that thinking about biology is very holistic. So the thing I want to say that so impresses me about Susan is something that I had to learn the hard way. So I started off life as a mathematician. I was at Princeton just minding my own business with Lonnie, and we were invited to Microsoft when they opened up their research division. And they said, one day computers will become really useful, but we have to gather lots of data about people and how they use them. And then, one day artificial intelligence will scale and it'll be useful. But between now and then, we have to gather all this high dimensional data and make something useful out of it. So we helped Microsoft start their early work in machine learning. But I showed up and I said to my friend who had invited us, so who am I? And what is my title? And what do I do? He just looked at me and said, well, we could draw a box around you, but if we do that, you have to live inside that box. I was like, no boxes. So Susan came and she had no boxes. She thinks about epigenetics, she thinks about cells, she thinks about humans. And it was just, it was such a learning experience for everyone in the lab to work with Susan. I'd like to say she has no boundaries, but I know to you guys that probably means the wrong thing. I would like to say her scientific interests and her enthusiasm have no boundaries. So I'd like to introduce Susan.
Thank you, Steve, for that very kind introduction. My name is Susan, and I'm very excited to tell you about some of the work that I've done here at UCSF. I had the fortune of joining this program, and this program being very flexible. So between my second and third years of residency, I took 2 years of essentially full-time research in Steve and Lonnie's lab. And a lot of the work that I'm going to share with you was either done or started during that period. So I'm very grateful to the program for that. So I actually started out my life in science as a geneticist. And as I proceeded in my training, both my scientific and clinical training, I realized that in psychiatry, there's what I perceive to be a paradox that I think we all grapple with, which is that on the one hand, psychiatric disorders are very highly heritable. Right? We hear these percentages of heritability estimates like 50, 90% for bipolar disorder, schizophrenia, et cetera. And many of the genetic risk factors seen in one disorder are also risk factors for another disorder. And yet, each of our patients presents very differently. And the question is why? And clearly that speaks to environmental influences that are at play. And I think we all see from firsthand experience that really what can set the course of illness is what stressors patients are exposed to and how they respond to those stressors. So for my postdoc, I decided to step outside of the world of genetics and study stressors. So I think one way to conceptualize disease is to think of it as an inability to recover from stress. So over time, right, we might have some baseline level of function, and then we encounter some sort of stressor. And if we decompensate and this decompensation persists, that's where we start to develop pathology. But that's not always what happens. We all encounter stressors all the time. And what often happens is that we might encounter this stressor and we might have a transient period of decompensation, but eventually we recover. And it's this process of adaptation that really enables us to maintain well-being. And I think a really good example of this is hypoxia. So for anyone who has ever gone to high altitude, you know that initially you have shortness of breath, you're feeling uncomfortable, but after a few days you acclimate. And actually mice also acclimate as well. So here are some mice in Normoxia happily roaming around. And when they're acutely exposed to hypoxia, they just kind of are in almost a state of shock where they're just not really moving around. But then if you give them some time, then after a few weeks in hypoxia, they return much to more of a baseline level of activity. And so this is one example of a phenotype, but there are many different phenotypes that we can measure. And this adaptation occurs not only at the level of the organism, but also at the level of cells. And I'll tell you more about cells later. But today, first, what I want to tell you is why I think hypoxia is really important in psychiatry, and to explore ways that we can go about understanding adaptation to stress, in particular hypoxic stress. And then at the end, I just like to spend a minute talking about how I think about like ways that we can integrate our understanding of genetic risk factors and environmental stressors. So as Steve alluded to, our lab didn't work on hypoxia before we joined, but we quickly realized that hypoxia was relevant to a lot of what the lab was studying, including cancer and neurodegeneration. And for me as a psychiatrist, I had particular interest in hypoxia for several reasons. One is that hypoxia-related diseases, such as ischemia, they have psychiatric manifestations, right? So after a stroke, you can have neurologic and psychiatric symptoms. After a heart attack, you can have like post-myocardial infarction, depression, for instance. And then also things like obstructive sleep apnea, we know have prominent psychiatric symptoms, such as anxiety and depression. And so these are hypoxic diseases that give rise to psychiatric symptoms. And our patients are especially at high risk for these for various factors, things like elevated rates of smoking. We use medications that have adverse cardiovascular effects and metabolic effects that put our patients at high risk for hypoxia-related diseases. And then thirdly, there's accumulating evidence that hypoxia in and of itself is important for neuropsychiatric disorders. So for example, in schizophrenia, hypoxic injury in the perinatal period is one of the strongest risk factors that we know of. And there's also growing interest in the neurodegeneration research community in hypoxia. So overall, hypoxia is relevant to a very broad range of diseases. It affects all different organs. And I think what's also important is that not only is hypoxia important for disease states, but it's actually also involved in normal development and normal physiologic responses, such as for wound healing. You need hypoxia as a signal. And so hypoxic responses are actually very highly conserved across evolution, and there's a really core hypoxia response. So how can we go about understanding adaptation to hypoxia? So we've pursued several different approaches. One of it is that we can survey what happens endogenously. So if you have an organism that's exposed to hypoxia, how do they respond? And from here, can we identify mechanisms that might be adaptive? And so we did multi-omics profiling. So we looked at mRNA and protein metabolites. And sort of think of this as like different layers, like in Google Maps. This is Byers Hall, by the way. Come visit us sometime. And we can really take these different types of information and overlay them to get a landscape of what the response to hypoxia looks like in vivo. Another way is that we can modulate hypoxia, cellular hypoxia adaptation. So we can take cells that are treated with acute hypoxia and expose them to drugs and see if we can mimic a chronic hypoxia type of phenotype. So in other words, we are trying to mimic adaptation, and from there, we can infer what are the innate mechanisms of cellular adaptation. And a third way is for us to directly dissect out the circuit of components at the molecular level. And that's going to be the focus of my talk. But first, just to tell you about what we already know about hypoxia pathways. So hypoxia, cellular hypoxia has been really extensively studied, largely due to interest in hypoxia in cancer research. And so here's the textbook version. There's a transcription factor HIF that is hydroxylated by enzyme PHD. So PHDs are oxygen-dependent enzymes. They use oxygen as a substrate to hydroxylate HIF. And this hydroxylated HIF is degraded. And so actually under normoxia, you have a constant turnover. You're making HIF and breaking it down. But when you have hypoxia, you no longer have this oxygen. And so this enzyme can no longer do its thing, and you actually get an accumulation of HIF. And so with this buildup of HIF in hypoxia, it translocates into the nucleus and turns on an array of target genes. These include genes that are involved in the delivery of oxygen, so things like angiogenesis, red blood cell production, as well as a metabolic shift towards glycolysis away from aerobic metabolism, and many other responses, some of which we haven't really characterized. But at the core, what we see is that we have a stressor, in this case, hypoxia, and there's a sensor that leads to a downstream response. So that was the textbook version, but in reality, there's actually a lot that we don't know. So I mentioned that we have this canonical sensor PHDs, but there are actually about 200 oxygen-dependent enzymes, and in principle, these could be sensors as well. And although there are several dozen well-known HIF target genes, there are actually hundreds, if not thousands of genes, most of which are not characterized. And there was another postdoc in the lab, Dr. Lily Lee, who she had a background in medicinal chemistry, and she was really interested in these oxygen-dependent enzymes. And I was really interested, with my background in gene regulation, in these target genes. And so we decided to team up together to see if we could identify novel sensors and key effectors involved in hypoxia response. And a couple of the challenges that we faced, one of them is that we're dealing with a large number of genes and a large number of potential phenotypes. And so we want to be able to capture a broad range of phenotypes. And this is where our lab's approach of phenotypic profiling comes in. So in this approach, we have cells that are in a plate, for example, a 384 well plate, and we can treat these cells with perturbations. These can be drugs or it can be genetic perturbations. Today, I'm going to focus on our drug treatments. So we're using drugs as tools to study responses. And in our hypoxia studies, we have a plate in normoxia, and we also have a plate that we've put into our hypoxia glove box. After treating these cells with hypoxia and our drugs, we can then use our automation system to help us do our staining with biomarkers and imaging. And then we get these microscopy images of cells that are treated with these different conditions. And for what I'm going to share with you today, we looked at a cell line called AC16. It's an immortalized human cardiomyocyte cell line that we thought would be a good starting place as a proof of principle. So once we have these images, we then do computational analysis. And I just wanted to go into a little bit about how we do this analysis because I think it's useful for looking at the data together. So in order to do this, we're going to zoom in at the single cell level. And what we do here is we use computer vision to extract single cell features. And what I mean by that is things like the size of the cell, the shape of the nucleus, the intensity of spots. And we can look at all these different features for every single cell within the well, and then we can compare across wells that have been treated with different drugs. So for each of these features, we can get a score, and we're looking at hundreds of features. So this is a whole lot of data. And to kind of wrap our head around this, we do dimensional reduction. Many of you in the room may be very familiar with dimensional reduction, but even for those of you who haven't heard this terminology, there's a good chance you're doing dimensional reduction on a day-to-day basis. So for example, we give these questionnaires to our patients, such as PHQ, depression questionnaire, and we ask them about these symptoms, and we give these numbers, and then we take these scores and we condense them down into a single score. So for example, you have a PHQ-9 score of 12, and we give that an interpretation, moderately depressed. So in an analogous kind of way, we can take our features and simplify them into these well-level profiles. We do a principal components analysis is our version of this dimensional reduction. And so each dot here represents a well, and we have a negative control population. In our case, it's a vehicle control. And what we're looking for are drugs that cause cells to change their response to hypoxia. So you move away from your control population. So now we have a setup for a focus screen. We have our hypothesis that among these oxygen-dependent enzymes, there are some sensors waiting to be discovered. And going through the literature, I curated a list of candidate HIF target genes that may be key to hypoxia response. So these are our candidates, and we've got our readout. And so now we need our positive controls. Sorry, I meant to say that. Among these candidate sensors, about a third of those are targeted by commercially available compounds. And for the positive control of those, we used a compound called IOX4. That's an inhibitor of PHD, which is that enzyme I mentioned earlier, that is the canonical hypoxia sensor. And so the pattern that we expect to see for a sensor is as follows. So you have your oxygen-dependent enzyme, and in normoxia, you have a high level of activity. It's happy, it's got this oxygen to do its thing. So if you give it an inhibitor, the activity drops. And what this looks like on our plot is that the drug causes a deflection away from the control population in normoxia. In contrast, in hypoxia, you no longer have your oxygen around, so the enzyme can no longer do its thing. And so it's got low activity. And if you add the drug, there's not much that happens because it's already low activity. And so in hypoxia, you don't really see much of a change. There's not much movement away from the control population. And if we take these data and we overlay them to each other, then we can directly compare the behavior. And so the pattern that we expect to see is that in normoxia, there's a big shift, so a long arrow, and in hypoxia, you see a small effect. So for a sensor type pattern, you expect to see a larger change in normoxia than hypoxia. And for a positive control, this is actually just exactly what we see. So these are the real data, and we see that there's a more dramatic change in normoxia compared to hypoxia. So that was for the sensor side of things. And as far as the effector size, about a quarter of these were targeted by commercially available compounds. And for our positive control, we chose a compound STF31. This is an inhibitor of a glucose transporter that's known to be strongly induced by hypoxia and is involved in this well-known switch towards glycolysis. So in the case of effectors, the pattern that we expect to see is actually the opposite. So in normoxia, that gene isn't doing as much, but in hypoxia, you have an upregulation of that gene. And so you have an increase in activity, which means that if you give an inhibitor compound in normoxia, there's not much that happens. But if you give this compound in hypoxia, then you see a more dramatic reduction in activity. So again, so this is the pattern that we expect to see, is that there's not much change in normoxia, but more of a change in hypoxia. And if we overlay the data, we see the opposite effect as for the sensor, where in hypoxia, we see a larger effect than in normoxia. And so for our positive control, as SDF31, that is exactly the pattern that we saw. So now we have our setup with our controls. And to summarize the work here, what we found for the sensors, one of our top hits was an inhibitor of KDM6, which is a chromatin modifier and also happens to be associated with autism. But I'm going to focus here on our top hit from the effector side of things, which was teleglenistat, a inhibitor of an enzyme called GLS. So teleglenistat had this effector pattern where there is a larger change in hypoxia than in normoxia. And this is in the high dimensional space with all of those different features that I mentioned. But if we were to just look at the raw images, what we see is that, so here's normoxia at baseline, just our vehicle control. When you treat these cells with hypoxia, you see an accumulation of these dots. And so here, blue is staining the DNA. And in green, there are lipids that are stained. We also have a mitochondrial stain overlaid. And what you see is this accumulation of lipids. And we know that lipid droplets accumulation is part of cellular stress response. So that was for the vehicle control. And if we treat ourselves with teleglenistat, this drug that inhibits GLS, we see that in normoxia, you do get a slight increase in the lipids, but in hypoxia, you get a more dramatic increase in the lipid accumulation. And so this is reflecting that we have this stronger effect on stress response in hypoxia from this compound. So what teleglenistat is doing is it's inhibiting an enzyme called GLS, glutaminase, and this enzyme converts glutamine into glutamate. And glutamate can then be converted into alpha-ketoglutarate and put into metabolism, TCA cycle, or it can be converted into glutathione to deal with oxidative stress. And right now we are doing some follow-up studies to look at these downstream branches of GLS effects. But overall, what we have here is we have a way from going from a hit from a screen to an interpretable phenotype and to generate testable hypotheses. So to summarize, we now have an approach where we can map components of a response pathway where we have many genes and many different phenotypes. And we are simultaneously in the same set of experiments detecting multiple components with different behaviors. And of course, here I've been talking about hypoxic stress, but this type of approach can also be applied to really any stressor and other types of responses. So that brings me to the last point I'd like to make, which is how do we go about integrating our knowledge of genetic risk factors and environmental stressors? So I think this is where we get into the realm of what I'd like to call systems psychiatry. I've been talking about wild type cells, but we can have cells with different mutations that are associated with psychiatric disorders. And I've been talking about hypoxia, but we can look at other stressors, such as environmental toxicants. So for example, many of the toxicants in cigarette smoke induce oxidative stress. And oxidative stress has a lot of overlap in response compared to hypoxia. There's a lot of overlap in pathways. And on top of all this, we can also look at the effects of medications. And so we can look at the combinatorial effects of genetic risk factors, exposures, and treatments. And I think this is much more reflective of reality, right, for our patients, because they're being exposed to many different things at the same time. And I've been talking about using immortalized cell lines, but of course we can go beyond that. We can use iPSC-derived patient cell types, and we've been starting to look at brain cell types that are iPSC-derived. And then going from 2D culture, we can go into 3D culture. We can do 3D organoids, we can look at brain slices, and we can of course also go in vivo. And so I've been talking about these readouts that we get from our microscopy images, but the same type of approach can be applied to all different modalities of data. So we can go from our molecular measurements to electrophysiology and behavior, and then even brain imaging. So I think this is the way where we can start to think about our biopsychosocial model of psychiatry, kind of bring it to the molecular realm. So we have a way to mechanistically understand the effects of stressors. And I think that this type of approach is how we can start to really bridge basic science research with health services research in psychiatry. So that I'd like to thank my lab and mentors and also my sources of funding. And I'm more than happy to answer any questions.
What drug library did you screen and how did you choose it? And if you had like a dream library that you could screen, what would it be?
Ooh, thanks, Linda, for that question. So for this hypothesis-driven screen where we were looking at our candidate sensors and candidate downstream effectors, we built a custom library. So we curated the list of proteins, the sensors and effectors that we were interested in, and then we cross-referenced it to databases like the Kemble database to look for compounds that target these. And in particular, we wanted compounds that had high potency. So we looked at things like the IC50. So that's what gave us our library for that particular set of experiments. But in terms of my dream library, so we did have an experiment where I'd mentioned there's this approach where we can sort of mimic adaptation. And for that particular set of experiments, we used a larger compound library, about 6,000, 7,000 compounds that includes many of like FDA-approved compounds as well as other kind of well-characterized tool compounds. But what I'm really interested in now, as I move toward this kind of environmental aspect as related to psychiatry, is I've been looking at environmental toxicants. So I have a library of several hundred environmental toxicants, and this includes pesticides and other types of pollutants and heavy metals. And I'm also interested in antioxidants. So like, for example, dietary derived antioxidants. So I'm in the midst of obtaining an antioxidant library that we can use as a counter screen. And I think that this dietary aspect is particularly important and also something that's very actionable clinically.
Thanks, Susan. That's a great talk and awesome work. Real quick question. I'm wondering, when you're considering screening your cells and adding in toxicants, how do you decide on, for instance, what concentration of toxicants to put on that might be either environmentally relevant versus super physiologic?
That's a great question. And I think, Richard, that the whole field of environmental health sciences grapples with this. So the question of, what doses do you use? What's relevant experimentally versus clinically in the patient? And I think that this is often honestly a consideration that is not addressed deeply enough. Honestly, a lot of times people will use what's out in the what's there in the literature or what's convenient on kind of the time scales that are convenient. But I think it's really important that we survey a broad range of concentrations, because you can have, especially when you're looking at combinatorial effects, non-additive effects such as synergistic effects or antagonistic effects can be very dose dependent. And so that's where you really need your matrix of compound number 1, all these doses, compound number 2, all these doses, and then you get this like exponential explosion of potential conditions, right? But I think that's a really important consideration. And also dosing is not just concentration, right? It's also time. So acute versus long-term exposures. And, you can have like sort of cumulative exposures that are low dose over time, or you can have sort of a pulse of high dose exposures. And I think that can reflect, say, like aging accumulation with over the lifespan versus maybe a more acute exposure during pregnancy in utero. So to answer your question, I want to do everything, all of them, but of course we have to be practical. And so I think like initially we really just have to cover a broad range. And I think right now the field has really focused on sort of short-term exposures, but I think especially as our in vitro models, our experimental models, brain organoids, and other types of 3D cultures, as they get better and we're able to maintain them longer term, then I think we can have more realistic models of longer kind of cumulative exposures.
All right, thanks very much to both of our speakers.